Well, this is the third part of our series of lectures on the history of genetics, at least the history of some of the areas of genetics that we're talking about in human systems. And so what I thought we would do is to now move into some of the actual techniques that are involved and where they came from and how they might play a role in our understanding of human genetics. So again, one of the reasons for this and to drag us through all this is that as we read papers, we'll find out that there are these techniques that have been used. Sometimes they're still used. Some of them are very functional even today that relate back. And particularly now, once we've got to where we can genotype highly efficiently, we're going back and looking at expression of particular genes. We're looking at the genetic code itself and relating back to things we already knew that were markers for various pieces of human traits. So I thought I would just work through a series of these, kind of give you some ideas of how they might work. I'm not going to go into depth on any of them in great detail, but I'd be happy once we get to papers or someplace, or if you happen to see some in your readings and you say, I don't understand how they do this, I'll be more than happy to Zoom with you and maybe illustrate how it might work, okay? All right, so we've already looked at the first one, which is visual mutants, okay? And visual mutants are things like whether I can roll my tongue, whether I can have freckles, or whether I have a widow's peak, and those. And you know, they're quite useful as information as far as how these morphologies might function. Okay. Now, the problem with all these is that they do have some genetic component, right? But they're not simple, as we pointed out in the earlier lecture. They're not simple little one locus, two allele problems, right? There is variation attached earlobes. There's fully attached and unattached and everything in between. And certainly there's genetic control. We see that if we follow pedigrees along, we see that there's a relationship with different lineages, but nowhere is it just simply a big A, little a, or big F, little f relationship, okay? But it doesn't mean that they're not important. It doesn't mean that we can't work through those and try to understand what the genetics is that follows them, okay? Well, the second thing we can do that fits into this, of course, how you do this really, is you don't just go out and count up the number of individuals that have attached earlobes or whatever. You tend to place it inside of a pedigree, okay? So this is a human pedigree, and actually we will, and after these history lectures, we'll talk about pedigree symbols and actually have you work some problems on pedigrees. We'll try to give you some more advanced symbolism than you probably had in some other courses, but we'll talk a little bit about that. But anyway, you work through pedigrees, you see who has it and who doesn't, and then you can figure out the mode of inheritance potentially. See if it's a dominant or recessive or whatever, okay? Other things you can do, of course, is that we get big pedigrees and you start understanding the relationship and how much information is contained. How related am I to my parents? Well, I get 1/2 of my DNA from one parent, 1/2 from the other, so 1/2. How am I related to my sibs, my brother or my sister? What's about 1/4 on average, right? Because we have independent assortment, we have all the chromosomes being mixed up, 1/2 and 1/2 is 1/4. And as we get further and further away from our ancestors, we become less and less related for us. But we do carry some of that information, okay? And it can be important to look at these forms. Just an example, you know, one of the places we really see these, again, I talked about the peerage and all the relationships of these families. Well, one of them that's very important is the Hasburg Dynasty, which ruled a great part of Europe for a couple of hundred years. And it was all started out by these emperors in Hungary and well, Spain as well, Castilla, who with intermarried. And the big thing here is that in order to keep the dynasty going, in order to keep this land over here, this country or this part going connected, you would marry somebody, you know, between the two. And this led to a huge amount of intermarrying, first cousins, second cousins, and a lot of uncle niece type relationships that were these forms. And it ultimately led to a series of really strong inbreeding outcomes. And we'll talk later about calculating the inbreeding coefficients, how inbred were the individuals, what's bad about inbreeding, what's good, okay? Actually, yeah, it can be good, okay? Almost all species have some level of inbreeding. But to this level right here, you end up with bad things, with the actual dynasty dying out with Charles II of Spain, who was morphologically very far removed from a normal human phenotype, long, elongated skull, had the mental capacity of someone maybe 5, 6, 10 years old, reproductively non-functional, not able to produce sire any children, and just died at a fairly early age. He was the last of the lineages, okay, in form. And there were lots of traits. You can actually go back and you can see this big classic chin that was found here. You can follow those back to where it came from Margarita back in here and Charles II. And so there are things that these morphological traits that you can actually follow if you can put the pedigrees together. All right, so visual markers. Well, they're easy, okay? That's one of the nice things about them, okay? Quick and easy to ask that. You look and you go, yep, they've got a roly tongue or they don't have a roly tongue. They've got, this portion or not. So they're fairly quick. They don't cost much. Yeah, you can just, send out a flyer, or you can just wander around, or people have sat in their cafes and, in Paris and counted people going by. They're looking at fluctuating asymmetry, which is, we know the right side is equal to the left side of the body. They had some traits they're interested in, and they would just watch people go by while they drink their wine and write down, okay, I had 10 people go by that were primarily left, 10 people that were right, 25 people that were this. So it's low cost, okay? It's directly related to, Darwin's ideas. That's what Darwin worked on. He didn't know anything about genetics. I mean, he struggled with that his whole career, trying to figure out a genetic, explanation, but he worked on variation, right? Variation was in morphological traits that you could see. His Darwin's finches with the beak sizes and the modifications there, the tortoises, his domestic animals that he worked with were all related. So it directly relates. Disadvantage, a lot of hidden variation going on, right? I mean, you got a lot of dominance, you got a lot of codominance or incomplete dominance, you got all kinds of things. So really hard to figure out until you do a big fancy pedigree that you even have even the simplest modes of inheritance work out. They're very difficult. And if you get into things like epistasis, or more complex modes, well, you really, it takes a huge pedigree and lots of time to even try to figure it out. And of course, the traits that we like to look at tend to be polygenic. Human eye color, multiple genes involved. Human IQ, multiple genes involved, plus environment, plus all sorts of things, okay? And leading to that, the differences can be subjective. I could look at you and say, okay, you have a high forehead. Well, compared to who? Me or you or the next person? Is that a genetic trait or not? So, it can be, some things very simple. You either have blue eyes or you don't, right? But then, wait a minute, I have hazel eyes. So when I feel a certain way, my eyes are green, and when I feel another way, they're kind of brownish. What's that, right? It's subjective. It can be quite subjective, right? And then obviously the range of variation is very limited. Tongue rolling, I can either do it or I can't, okay? Well, wait a minute, some people can do it more, a little less than others, but it's pretty much, within 4 or 5 different phenotypic groupings for pretty much any of these traits. So the variation, really, not a lot of it that you can work with. Okay, now that doesn't say it's bad. It just says it's that for some things it's been very, very informative. I mean, look at if we look at color morphs and industrialized Europe, when we had coal and things being used in the early part of last century, and we saw the famous Moth experiments, right? You had light-colored and dark-colored moths, and there were just 2 forms, and then you had, whether the birds selected a form or not. And it turns out it's more complex than that. But sometimes morphology type things can be really, really informative. You don't need much more. Okay. So what are some of the other markers? Well, besides visual, there are physiological traits. People said, okay, well, let's get more complex things. Let's look at things, like resistance to certain drugs or to certain conditions. We can look at tolerance. Okay, how much alcohol is it different if I can take to do a particular task or do something else, or temperature. I had good friends that were physiologists in the military, and they were constantly subjecting people to temperature and pressure changes and all sorts of things to categorize individuals. And of course, those can be clearly under some genetic control. Life history, right? Longevity, or how many progeny you produce relative to someone else, etc. Of course, in humans, that's not life history is not as good. We mostly look at longevity. Things like longevity, I mean, there's some really recent work out in genetics looking at some genes that are involved in longevity. This thing, you know, the idea that we might be able to look at cellular senescence, organismal senescence, and slow that process down. So we might live another 20 years or another 30 years in good health. Okay, all right. So what are the advantages? Well, you can increase the number of variants because you have a distribution. There's a tolerance of temperature from really low to really high, and then lots of things in between. You can have related to fitness. Okay, again, it fits back to this Darwinian ideas. Okay, if I'm in an environment where it's cold and I can tolerate more cold than this organism over here or this individual, and I'm likely to produce more offspring successfully, or my offspring are liable to live better, whatever. That's a fitness argument. Okay, that you can manipulate them, right? You can't really manipulate eye color, right? It's just what it is. So, you know, that's a physical trait, but you can certainly manipulate this. You can take individuals and you can put them in a cold environment or a warm environment or do things and see what the outcome is. The disadvantages, well, as soon as we move down this chart or this list, we see that it takes more expertise, okay, to go, you know, you got to hook up equipment, you got to have more technology, you got to be able to understand that, the relationships and work with that, and it can take a lifetime to be a really good physiologist to understand even a few traits that you might be able to work with, a few markers. Most of the things we're talking about is also polygenic, right? Multiple genes involved, life history, how long we live, how many kids we produce, all that has a huge polygenic effect. And anytime you do this, you know, we're talking about a strong environmental component. If you think about, you know, my tolerance to cold or my tolerance to heat, well, you know, if it's in the summer and I've been outside working a lot for the last week or two, then you put me in, stress me with heat, I probably can deal with it better than I could if it was, say, January and I had been living, you know, in Wisconsin and it was cold, and you suddenly, you know, your physiology changes, your expression of genes, you know, that control your body's ability to deal with extremes of temperature, heat, and water, etc. Will change with your exposure. So there's a big environmental component to it. So given those, people started really looking at, okay, what else can we get to? And can we get away from the environment a little bit and really get down to the genes, to the genetic material? Well, where is the genetic material stored? It's stored in chromosomes, right? So people started looking at chromosomal variants and how those chromosomal variants might function. You can look at lots of things. You can look at just the arm lengths, right? Whether it's short arm, long arm. Is my arm short, you know, same form? You can look at just gross morphology, you know. Now in humans, we all pretty much have the same system, but if you want to compare one species to another, you can compare whether this one has 22 pairs of chromosomes or 21 pairs or whatever, whether this chromosome 11 is fused with chromosome 12 to make a different, you know, one hybrid or whatever. And then of course, in the late 1960s, they came up with ways to band chromosomes, to make them show up as light or dark areas. And we know that the dark areas happen to be heterochromatin. They're areas that pack up more dense during mitosis, and the lighter areas are euchromatin. So these are areas that tend to have more of the genes, active genes, not active obviously in mitosis, but once they open back up into G1, these are regions that are more active. So the banding pattern. So you can look and say, okay, this individual over here has this band and this one doesn't, or vice versa. And okay, and that became very, very important for looking at major, you know, at chromosomal systems. And we're going to give a whole lecture series here shortly on chromosome morphology and chromosome variation, just to remind you about that. But it was really important. You know, you could just count up how many chromosomes individuals had. Obviously, this one's got an issue because they've got 2 X chromosomes and 1 Y. Right? Can you remember what that is? Okay, 2 X chromosomes and a Y. Okay. Well, it's a male, right? Because it has a Y and it's got 2 X's, but it's got that. This would be a Klinefelter syndrome, right? And so we're just counting up the number of chromosomes and seeing that we have an extra sex chromosome here. So we've got a Klinefelter. So we know that this individual is going to have certain phenotypic expressions likely to occur based on the syndrome. But we could also go in and look to say, well, there's band right here and there's not over here. Okay? maybe that's been a deletion there. Is that important? Well, we look at the individual's morphology and we see that maybe they don't have any hair, or they don't, or their thumb's messed up or something. And we know that, well, the genes that somehow that are involved with that deletion, that missing band over here, must somehow be associated with, the development of the thumb or something else. So we could use them various ways. Now, the advantages, they are closer to the genome because you've taken away the environment. They are really good markers for systematics, for distinguishing species apart. Okay, so we're not talking about humans here, but for species identification, because they really relate to reproductive isolation. If I've got 22 chromosomes and you've got 40, and we try to pull those together, the hybrid's not likely to function, right? Even though we could physically combine our genomes, they're not going to be most likely be a viable progeny for that. It does tell us information about the entire genome size and its growth structure in various ways. Okay, disadvantages takes a lot of expertise. Okay, and living tissue is required, usually a mitotic tissue. So it's a mitosis base because we want those chromosomes when they're in the most condensed form so we can actually see them. At least that's the old way we do it. And so, we had to use living tissue. And there's little population level variation that goes on. And it does take a lot of expertise. I don't tell the story very often, but I went through we were doing this in Hawaii with fishes. And in our hotel room, we had all this equipment. We had dewars, so liquid nitrogen. Of course, we had to have syringes to inject the fish with cultracine. We had microscopes, we had slides, and we'd been off in the field, and we came back, and there was a tap on the door of the hotel room, and there was 2 police officers there with their pistols drawn, and this poor little scared guy that was the hotel manager, because the cleaning staff had come in and found all this, these chemicals and stuff, assumed, I guess, we had a meth lab going or something. So we had to, it took a little while to explain. So yeah, we had a little bit of fun in the middle of Hawaii, basically having all this required technology stuff with us. Fortunately, we had all the permits and we had connections with the Fish and Wildlife Service in Hawaii and all that. So it was no big deal, but it was a interesting moment with me answering the door and with my beer in my hand. Okay. All right, so now what? Okay, so we've got chromosomes. They're really good. They're still used today. We do a lot of chromosome variation studies. That's one of the things that first things they pull out when they do amniocentesis is to look at, just count the number of chromosomes, and then they start looking at variation to make sure that things are good. The next system that came along was really looking at biochemical variation, okay, proteins. What brought this along was the development of what is known as gel electrophoresis. Now, it started out as not gel electrophoresis, it's chromatography, where you just take some biological material, you let it pull up through some paper, and then you look at it. But we learned pretty quickly that if you applied an electrical field to it, that you could move proteins. And remember, proteins made up of amino acids. A group of amino acids have charge, right? Positive or negative charge. So if you mutate back at the DNA level, you're likely to change that charge by changing one of the amino acids. So if you happen to knock out, glutamic acid or something and put in a neutral one, then you're going to be more positive than the others. And so you put them in electric field, and those proteins will move at different levels. And this is known as allozymes and isozymes. Allozymes are the variations from a single gene in the protein that's produced. Isozymes are variation in different genes, different copies, okay? And they're separated on peptide charge, okay? What brought it about, first we could just do it with hemoglobin and things that bright red anyway, you could just see it, right? Then people developed stains. They looked at the biochemical pathways, and they said, oh, okay, I realized that we've got malic enzyme, okay? Well, it's an enzyme. It works on a substrate and it produces a product. So if I throw in the substrate and I let it sit there in the right conditions, it will, and give it some energy and stuff that it needs, it will do its little job and it'll produce the product. And in doing that, it often spits off hydrogens, okay, or something else. And there are chemicals that will grab those hydrogens and then just fall out of solution. And so you end up with taking these gels, and you can end up with little bands that represent where that protein was in this electrophoretic field, and you can get variation. Now, it's done all sorts of ways. So originally, Most of it was done with what was called starch gels, starch gel electrophoresis, because it was cheap and fairly simple. Um, years ago, you could go to the grocery store and you could buy starch. It was just starch as potato starch, you made from potatoes. And what it was used for was to starch shirts, right? So you'd boil it up on the kitchen, on the stove or something, and kind of mix it up the right solution. As soon as you finish washing your shirts before they got fully dry, you put them in there and kind of wring them out, hang them up, and they would, they'd be starch collars or starch shirts. Well, it turns out you could take some of that same type of starch, potato starch, and hydrolyze it, make it a little bit nicer. And when it hardened, came back into its solid form, it was fairly clear, right? And that's what these are. You can see through that. That's a layer of potato starch. So what you have to do is go into the lab, put the potato starch into a Erlenmeyer or Florence flask, and with some buffer, right? Because you got the right buffer, and you would just shake it. Like this over a flame. That's what this lady's doing here. You just keep shaking it over a flame, rotating it. So you don't want it to burn, otherwise it just turn black, burn. But you keep going. Ultimately, big bubbles would start to come up and it start to clear. Okay. If you're really good at it, you could do it both hands. You could do 2 at a time, and things. And then as soon as you set it down, pop a vacuum on it and degas it, and all the gas would come out of it and it would make us really perfectly clear little starch part. And you could pour it into these little molds like this, and now you had a substrate to work with. Okay, what you could do then is you could take some samples, biological samples. They could be blood. They could be it's called find them and grind them labs. You would take a whole fish, or you could take just a liver out of something, or an eyeball, or whatever you want. You grind it up in a buffer. You stick a piece of paper in it, okay, just a little thing, and you line it up, the samples up, and in this gel, and push it back together. And now you have you have all the proteins that, or at least the majority of them, that were in that organ or in that organism. And you could do it with trees. I mean, we did it with trees and cacti and everything you think, every kind of organism. Fine and grind labs did all sorts of things. You then put it in an electrical field, okay? You could use like these just wipes. I mean, it's a fairly simple system. Just about anybody could do it. With a buffer on one side, a buffer on the other side, a little electrophoresis unit. You could even do it with a battery. You may have done that in a genetics lab where they'd have you electrophoresis stuff using just a 9-volt battery, but a little more sophisticated. We do it in a cold room. And again, remember that you're just looking at these acidic or basic changes, and when they would come out, you could look at them. Now, what we would do, what's nice about this, now, many of you probably in cell lab and things have run polyacrylamide gels, either tube gels or flat gels. Okay, that's it. You got one gel, and you can't really do anything to them except, I mean, they're very useful, but you've got one gel. Okay, you may have different sections that you use, but starch gels, you could actually take essentially a piano wire and a little holder, and you could just pull it, and it would slice it. And if you're really good, you could slice 5, 6 slices out of here, which meant that you had these layers that you could actually use to look at 5 or 6 different enzymes. So you take the top layer, you might look at malic enzyme. Take the second layer, you might look at malate dehydrogenase. Third layer might be lactate dehydrogenase. Anyway, you could have multiple runs, and you lay them out, and then you would pour on this substrate. So you'd have, in this mixture would be whatever substrate you thought malic acid was going to work on. There would NADP or something to have it some energy sources, and then something to capture any of the electrons that would be released. Okay, and you like this hydrogen right here that's coming off, so we would throw in NAD here, NADH, form would beef out, and then you put them in an incubator about 37 degrees kind of temperature, let them sit for a few hours, and when you got finished, you'd have some blotches, okay? And those blotches then could be read as, either homozygotes or heterozygotes or something else, right? So here's a heterozygous individual right here. So he's got one protein form on one chromosome is here. The other one's chromosome had a different mutation, so you get a different response. So you've got heterozygotes like that. There's other kinds of heterozygotes, and this. After looking at these for a long time, you get better and better and better at reading them. This is a heterozygote as well. This is a dimeric protein, so this is a monomeric protein, so it only has one form. So whatever this chromosome puts out, you see that that protein product, whatever this chromosome puts out, you see that protein product as well. And then when they show up in there, they're together. Okay? But if you have a dimeric protein, sometimes you get AA together, sometimes you get BB together, and sometimes you get AB together. So you end up with 3 bands. This is a dimeric protein. And then there were tetrameric and pentamerics. I mean, so, you had to learn a lot of biochemistry in order to do this. But it let us genotype without environment things. I know that's a heterozygote right there. And I know it's different from this heterozygote over here. And that's different from this homozygote that's right here. And that's even a different one there. It's not, it looks like it's somewhere in the middle. So I would, if I were scoring this or I go maybe AA, okay, this homozygous AA, that's homozygous BB, homozygous BB, AA, AC maybe, okay, something like that. AB, whatever, okay? And you could genotype the individuals, and you could do it for lots of different loci. So now you had genotypes, had real structural genotypes. Now you could get pretty complicated. This is esterases, and you get all kinds of weird bands, and you have to learn. What I'm really scoring is just this stuff down here in this layer, and then that's a whole different part of the different form of the protein. Because remember, when particularly when you use whole organisms, you've got proteins that are in the eye and the liver and the kidney and the muscle, all the same basic proteins, right? Because we only have about 20,000 genes, but we've got hundreds of thousands, 100,000 plus proteins. So those have to be reused in slightly different forms. So malic enzyme that's in the eye is completely different. It's the same gene, but it's completely different modification than the malic enzyme that's in muscle. But because they're the same size proteins for the most part, they kind of sometimes overlap each other and look like this. So that was a pretty complicated thing. So proteins, what are the advantages? Well, you get a large number of loci. Eventually you could get a whole catalog. There's a book, famous book, that had recipes, we call them, for humans. And there was like 150 different proteins that you could look at in humans, 150 different markers. That's pretty impressive if you wanted to do it. You can use it for almost any plant or animal. And gosh, we did it on everything from deer, turkeys, to cypress trees, to just about anything you can think of. Okay? They're generally codominant, right? So heterozygotes, for the most part, can be recognized. You can see them. Okay? Disadvantages, they only survey enzymatically active proteins for the most part. I mean, there's some that could do esterase or could do hemoglobins and things, but most of it required that it be enzymatically functional. So, we missed a lot of the proteins that aren't enzymatic per se. They're linker proteins and things. Coding proteins are a tiny part of the genome. Remember when we did that little stuff in genetics, we say, okay, here's the cut curves and how much of it is unique and how much of it is highly repetitive and moderate repetitive. And oh, by the way, the unique part is only a few, 5%, or depends on the book you look at, 2%, whatever, of the actual human genome. It's not a very big part. So 90-some% of the genome couldn't be looked at with this technique, right, where a lot of the variation is. And you have things like null alleles, because you'd truly be a heterozygote, but this protein just didn't work at all. So you look at it and you think you got a homozygote, but really it was a het. It was just that this protein was this side, this chromosome that had produced this part of the protein was non-variant. And there was all sorts of other things that went on. So then what we do? Well, we said, let's go and get away from the proteins and go to the actual genome itself. And this was where things like restriction fragment, link polymorphism, RFLPs came into. This came along from the development of restriction enzymes. You may remember back from genetics when we talked about restriction enzymes have been found back in the 40s, and so it was one of the things that helped us understand about Griffith's experiments, right? Remember Griffith's experiments where they were trying to figure out whether DNA or protein or whatever was the genomic material. And they came along and they developed these ways of killing off proteins in the 40s, killing off proteins and cutting up, messing with DNA and RNA. Well, that was because there were restriction enzymes and other proteinases that were discovered and were available. So there were restriction enzymes. There are just hundreds of them, right? They're just little enzymes that chop up DNA, right? Why? Well, because they're naturally occurring, right? You get all this transcription going on. You got all this DNA floating around in the cytoplasm. You got to do something with it. You want to reuse it. It's a very, our biological systems are very efficient at reusing stuff. So what do? You chop it back up into its little pieces and resort it, use the DNA, use the message or the DNA or the RNA again. Okay, it could be RNA obviously in the cytoplasm, but you allow you to use those particular nucleotides again. And what this would produce is something that was called DNA fingerprints. It looks something like this. Okay, again, this was used on a polyacrylamide gel and forms. Now, what really you did was this: you just took some DNA. Okay, you had to get pure DNA, isolate it. Took the restriction enzyme, so you just get whole genomic DNA, right? You just take a bunch of tissue or something, and you get whole genomic DNA. You throw in the restriction enzymes, and they go to a specific spot. So these restriction enzymes would only go to a place where you had ATTTCG, okay? And if they did, they would cut it, nick it, right? And those of you who've taken recombinant lab and stuff know all about restriction enzymes and how you use them to insert vectors and all that sort of stuff. Once you nick the DNA, you could electrophoresis it. Okay, we would often do what was called a southern blot because you couldn't really stain it. You couldn't really see anything when it was in the polyacrylamide. So you would transfer it over to nitrocellulose or some other film. That's what this little big picture is over here. You would take some buffer, you put the gel above it, and then you put a bunch of paper. You put the membrane you want up here, then you put a bunch of paper above it, and wait, and literally you just suck the DNA slowly by osmosis out of the gel and into this. It would get stuck in this hybridization buffer membrane right here. And once you had that, you could then take it and work with it because the DNA is stuck, right? And you'd know you were around a hybridization lab, a southern blotting lab, because you went into any of the bathrooms. There was never any paper in the hand cleaning thing because you just use tons of this paper to stack up, to pull this out. And then there were vacuum ways, there were other fancy ways to do it. But for most people, and still today, people do southern blots, and this is one of the ways they do it. Okay. Once you had that, you would throw in a radioactive probe onto the membrane itself, or you could use a light probe, luciferase, whatever, some probe that was specific for the DNA that you were kind of looking for. Expose it to X-ray film and you'd eventually produce an autoradiograph. So that looks like this. Okay. Now, what was interesting was this exposure to film, depending on how well your probe worked. It could be a few hours or it could be days. You can sometimes take you time to do all this, because it took you a while. This run, big old long run, often overnight. Southern blotting would run overnight at least, okay, for the most part. Probe on it, you probe it for a few hours in right conditions, and then you put it down in an ultra cold, so minus 80 degrees Celsius freezer, and let it sit, and it might sit for 3 or 4 hours. It might sit for a week, just depending on how much, how well your probe was and how much DNA you actually had. Then you take it out, take the X-ray film just like you would, when they do a dental X-ray or whatever, and expose it. And what you would get is what's known as a DNA fingerprint. Now you could do it with two sources of DNA, two sources of DNA for humans. You could do it with mitochondrial DNA, right, circular, and that was really good for things like looking at species level variation. So obviously humans, species, but if you want to compare that, you could get some nice bands, and that you only got 16,000 bases, so you wouldn't cut it but 4 or 5 times, and so it wouldn't be so messy. If you took whole genomic, depending on the probe that you used, you would get hundreds or dozens of bands, and you could really end up. Now, these is called DNA fingerprint. The thing that's different here is that, remember, you're just cutting up the DNA, so the presence or absence of one of these little bands is binary. It's either there or it's not, right? For example, this band right here is in this individual, but it appears to be missing in this individual. That's not a het. That's not a heterozygote and a homozygote. That's just a missing band. So it became a binary system. So it was ones and zeros. So mathematically, that's far more complicated to work with than it is if you're trying to deal with, classic Mendelian 2-allele system or presence or absence systems. Okay. So what are the advantages? Well, you got a large number of sites in your genome. The restriction enzymes are all over those chromosomes, so they hit this number 1 in 20 places, number 5 in 14 places, number 2. They are markers for actual mutation, because if there's a mutation in the region where the cut's going to be, it won't cut, or it will cut. Okay, so you end up with real markers. They're highly variable. I mean, you just get, all sorts of individuals, right? It requires a lot of DNA, a lot of pure DNA, good DNA. It's laborious. It takes days to weeks sometimes to get a result working with it. And in its presence or absence again, this either your band is there or the band's not there. You couldn't tell if it was a het or a homozygote. That was really limiting to some of the existing theory. Then once that was going on, this was a really technique from the 1980s. We had the development of PCR, and almost all of you know about PCR now. You take unamplified piece of DNA, you heat it up so you get it into single-stranded. You throw in a primer, a piece of DNA, a little manufactured piece of DNA that recognizes a section of that single-stranded, locks down, and then you basically just do DNA replication in a test tube. So you throw in all the things you need: RNA polymerase, a bunch of trinucleotides, ATP, GTP, CTP, and the right buffer so it's all happy and everybody's in good shape. And you cycle it. You go heat it up so it pulls apart, cool it down, let it do all of its little things it needs to do to replicate, heat it up, put it out, heat it up, put it out. And when you do, you end up with thousands and to millions of copies of that little stretch of DNA that you're interested in, right? So, and you could do it from anything, right? We do it. This is the primary technique that's used nowadays to, when you pull out any material, it can be a stain, it can be a blood sample, it can be hair, it can be long bone, it can be from teeth, extracted in mummies, it can be from all sorts of things that you can get it. These microsatellites are interesting because they tend to, again, have this pattern of heterozygote and homozygote, and so they tend to be patterning that really follows this codominant mode of inheritance. For example, this one, just ignore these little secondary bands. That's just part of the process when you run gels. This is a homozygote here, and this is a homozygote here. And then these individuals have both bands, so they're heterozygotes. So homozygote, 4 homozygotes. This one different, and then these individuals over here having heterozygous condition, just kind of very much like the proteins, except we're right at the DNA level. We're not worried about it being an enzyme or something else, okay? And of course, by now, most of you know how it works. If you don't, I'm happy to talk about it, but you find a repeat region, like this is a DNA sequence over here, and just ignore that stuff. This is a repeat region of, looks like GT, 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 so a dinucleotide repeat. We have those all over our genome. We have trinucleotide repeats, ATG, 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 or whatever, all over the place. And what you do is that you build a primer that fits in up here somewhere above it, and you build another primer that will work down here somewhere. It's complementary to this part, put in the forward and reverse. And so I can always go into a genome and find that little section. And because these are dinucleotide repeats, they often misalign when you go through replication, and so they tend to grow and shrink. Okay, and so they tend to sometimes add 2 and sometimes take away 2, and it's usually in 2s. Okay, and because they're 1 or 3s, if they're trinucleotide, because it doesn't line up properly, it messes up. Okay, so in replication, you get extra parts of it. Okay? Now, when you do all this, of course, you end up with a nice pattern that's used all the time. You've played with it in probably labs very much, where you get heterozygotes. These are really different heterozygotes. This is mom in this case. He's a heterozygote for these 2 very different forms. This is child 1 or child 2. Okay, this is dad. Okay, and this is child 3, and this looks like this is child 4. Okay, and sure enough, dad has contributed to what? Okay, dad's contributed this one here, right? Child 2. This one to child 3, this one to child 4. Mom's contributed this one to child 4, this one to child 2. Looks like she's contributed this to child 3. Okay, so and then 2. So everybody makes sense. Ooh, wait a minute. What about child 2? Okay. Well, child 2 matches mom, but what about dad's contribution? Dad had to give them the blue ones, either that one or that one. There's nothing here. There's nothing here. Okay. So it looks like child 2 didn't come from mom, yeah, or excuse me, didn't come from dad. So, it's one of those cases of paternal testing would work. We'd say, there's pretty good evidence here. Now you do, this is exclusionary, and you'd say, okay, we're excluding dad, the male, for this particular locus as being a potential contributor as the paternal parent, right? And you come back to it. Now, it could that this is uniparental disomy, which has happened to people. People have gone in and they've looked at a locus, and mom's is all there, but dad's isn't. And they go, ooh, and they come back and go, no, it turns out to be uniparental disomy, which we'll talk about later. But, it's the way it works, okay? Now, with microsatellites, to kind of finish that up, there are lots of other combination techniques. There's called rapids. Rapids is where you made up a random bunch of different little little primers. You could buy them for $10. You could get a whole vial of primers. Just throw them at the DNA and see if they cut. It's called rapids. Worked well in plants. Never worked very well for mammals or for humans. Another one was what you could do was single site partition, where you would take a gradient gel. This is a denaturing gradient gel, and you would pour it and you would amplify it up, and this would look for single mutations. And so if you saw, you could run it through this gradient gel. So at the top it might be not very stringent gel. At the bottom it'd be highly stringent gel. So it's really causing that DNA to pop apart or something. And if you saw variants in there, those would represent mutations. You don't know what they were. Okay, then you knew they were just somewhere in that stretch of amplified DNA, but you could look for them. Okay, and then there's things like CAPS. CAPS was a combination of PCR and RFLP. You would amplify up a region of say mitochondria, and usually what you did it with, and then you would take a restriction enzyme and put it on there, and you would see somewhere in that region, because you're not looking at repeats now, right? You're just amplifying up a big portion of the mitochondria. So you got real sequence, not a lot of repeats. And you throw in restriction enzymes on top of it, you see where it cuts. If it cuts in one place, one time, and it doesn't cut in the other, then you've got a mutational difference. You don't exactly know where, but you got it. I threw up these little fish up here because we did this years ago. Northern Alabama, north central Alabama, they have both white crappie and black crappie. They occur in the same reservoirs, and they were finding hybrids. And the local folk, fishing lore was that it was always one way or the other, right? It was always a male black crappie and a female white crappie when they spawn. So female would lay eggs, and the black crappie would there. And so people, I don't remember which way it was, but there was a lot of folklore about it, and people were really interested in it. So people over in fisheries started collecting these and the hybrids. And we started looking at it. Well, they did allozyme work. They did protein work. Okay, so they could look at the nuclear markers over here, and they could see that they were hybrids. That's one of the ways they could do it. And then we did the mitochondrial work. We took RFLPs of some PCR for fishes and looked at it. And so the question was it always a male? Because remember, mitochondria comes from the female, right? That's where we get it. So was it always a white crappie? If you took a hybrid fish, was it always a white crappie mitochondria that's there? That meant the mom was mitochondrial, was the contributor there. Or was it always black, which meant that the female was a black crappie, or was it something else, right? In the form. And so they would take the allozymes and they would find us these hybrid fishes, and we would isolate the mitochondria. We would do the little PCR and the RFLP with it, where we knew it worked, and we did. And so what we found out was, guess what? It was basically equal to the frequency of the two fishes in the thing. There was no directional, hybridization. It wasn't always a black crappie male and a white crappie female, or vice versa. If you had 40% black crappie in the lake and you had 60% white, then the hybrids about half the time or about 40% of the time were showing that mom was a black crappie, and about 60% of the time were showing that mom was a white crappie. It was a relationship. So it's kind of fun. Something we did, using sort of these little mixed of techniques. All right. So what's the last of these? Well, we'll try to move this on, is nucleotide sequencing. That's the biggie, the biggie today, right? Where we can go in, we can look at the actual sequence that's there. Now, for years we had to do this. I mean, it was a laborious process. When I started sequencing back in the 80s, you know, you would take DNA, you take it, you would put it in a vector. So you would isolate the DNA, you cut it up with restriction enzymes, you put it in a vector, grow it up in bacteria, isolate the DNA from the bacteria, give it pure DNA, you would put the sequencing reaction on, you'd run a gel for hours and hours and hours, take that gel, put it down, you know, what you would have marked it, the DNA with radioactivity, put it down in ultra cold for days and days and days and days, and eventually pull it up. That whole process to get a few hundred bases, you know, like this one little gel right here, take about a month. And a month of time of lab work, and sometimes 12, 14-hour days just to do the whole process and get it right. Okay, we did it, you know, the first time we did it, we worked and worked and worked for about a month, finally got our first sample. And back then, instead of going to GenBank or Simpler Online, this is in the 80s, before a lot of the internet was really active, you'd have to go over, somebody had a big disk drive with it stored all the data that was, which wasn't very much, stored on a disk. Once a month, they'd get a new disk shipped in from Washington, and we'd go search, and you'd have to trying to search, you know, on this old computer. And the first time we did it, we got a bunch of sequence like this, and we coded it up, and we ran over there, and we threw it on. And it was like, you know, searching all night long. Next day we came back, and there was our little printout, and we were working on Gambusia on live-bearing fishes, and we looked at our sequence, and it was 99.9 human. So after all this month of work, we somehow managed to contaminate it and sequenced ourselves. We got a little better at it, but anyway, of course, nowadays, you know, you do it with automated sequencers, and so you see the different colors mean different bases. And then we can even do multiplexing, so that within one lane right here, instead of having to run one gel with 4 lanes for each one and all that, each one of these lanes is one piece of DNA, and the technology, you know, for now, we can sequence an entire genome in 48 hours or even less. So it's so, you know, the new technologies, we can get a single fragment. Of course, we can do mitochondrial sequencing, which is really good. We can use the whole exome. So just focusing down on only those pieces that are being expressed. Okay. We can look at transcriptones, which is really fun and really good. You go into a cell, and now you sequence the RNA. Right, figure out what messages are being produced. So if I look at this, if this individual's got this particular disease, and I go pull some cells from that and look at the individual, are they having a different expression pattern than somebody else? So maybe they're heart disease and this one doesn't. I can do the comparisons. And of course, whole genomic. Now, you know, unheard of to be whole genomic, but now we can certainly do it very quickly. You can get yours done for, you know, hundreds of dollars now, maybe thousands. Of course, big question is what you do with all this data, right? You got a huge amount of data. It's not trivial to sort through it and look for things, but you can do it. And there's even single cell. We'll talk about some of that later, but one of the things that's used in reproductive human technologies is that you will grow up you'll take a cell, you take an egg and fertilize it, wait for it to do a few cell cycles so that it's in mitosis, so that you've got, you know, blastular or maybe a more or less stage where you've got enough that you can take out a cell and not hurt anything. Take one cell out and genotype it and tell you, you know, exactly what it is, so you can see whether you want to implant that one or not. So that's single cell sequencing. Okay. Now, last thing to say about this long lecture, sorry, is that if you look at it from a standpoint of all these things together, the amount of information at the top here, morphology is low, right? A few markers, a few things. The amount of information from nucleotide sequencing is just massive, just huge amounts of information. But the technology required to do those two things is really different, right? Up here, we can do it. You know, sequencing is, I will say the technology has gotten easier and easier. Okay, I can go in, I can amplify something, put it at 96 well plate, ship it off, and, you know, a week later have some really beautiful sequence out of it, which I couldn't do years ago. But it still, you know, requires a lab and understanding and thermal cycling and all the technology to do it. But so just a basic little kind of idea of what's going on. All right, well, that ends what I wanted to say about that. I just wanted you to at least have an idea when somebody said, I'm doing protein electrophoresis or I'm doing RFLPs. What the heck are they talking about? Just some way to be able to kind of put that, in your head as we talk about it. All right, and the last of these history lectures, the next thing I'll talk about are just some examples of human genome programs that are going on. So now that we can sequence, what are we doing with it? We're interested in human genetics. What are we actually doing with this system?